All right. Welcome everybody to today's UCLA CTSI Distinguished Speaker series for the Regenerative Medicine team. It is my distinct pleasure to introduce today Dr. Elana Fertig. Dr. Fertig is a professor of oncology, applied mathematics, and biomedical engineering at Johns Hopkins University. She also serves as the director of the Division of Quantitative Sciences and the associate director in the Division of Quantitative Sciences at the Sidney Kimmel Comprehensive Cancer Center. She's also the co-director of the Convergence Institute and of the Single Cell Consortium at Johns Hopkins. And this truly emphasizes the role she has in spearheading transdisciplinary research projects that bring together experts from very diverse fields to tackle complex challenges in oncology. Dr. Fertig is a leader in computational oncology who integrates systems biology with translational approaches to advance new predictive quantitative medicine paradigms. Her transdisciplinary expertise enables to lead large-scale team science projects that adapt cutting-edge molecular profiling technologies to human research to uncover new therapeutic interception pathways. She, as I mentioned, is a leader in computational oncology and has a very interesting background as a NASA research fellow in numerical weather prediction theory, which I believe has uniquely positioned her to apply this type of complex data simulation techniques to cancer biology. And in fact, some of the very interesting, projects that she has spearheaded and developed includes applying computational techniques to truly forecast cancer progression and therapeutic resistance, in a fashion akin to weather prediction, which I think she's going to tell us a little bit more today. She's a pioneer in applying spatial multi-omics, matrix factorization, transfer learning approaches to the study of cancer, and really has established these as, methods and state-of-the-art in computational biology. Her contribution have been recognized by, many accolades, including her election to the American Institute for Medical and Biomedical Engineering College of Fellows in 2022, and she has also been, nominated as a Maryland Moonshot Senior Scholar from the Sidney Kimmel Comprehensive Cancer Center in 2023. She has a very extensive publication, record. She's a leader in the field, and she is training the next generation of computational oncologists. It's, just a fantastic person, which is doing some groundbreaking science, and I hope you will enjoy her talk. Thank you so much, Elana.
Thank you so much for the kind introduction, Alice, and, for having me here today. It's a real honor to be here, albeit remotely. And please, even though it's remote, put questions in the chat, and I know Alice is going to be monitoring it during the talk. And, I'm happy to take questions as we go, as well as at the end, as much as that works for the Zoom platform. So let me try to share my screen.
And while Dr. Fertig shares her screen, yes, she's completely right. Please do use the Q&A panel, which I should have reminded you, to ask any questions, and we'll find a way to get them to Dr. Fertig. Thank you.
Great. So I'm going to talk today about some work we've been doing on forecasting pancreatic carcinogenesis as we think about using spatial multiomics technologies. And briefly, just my disclosures. So it goes without saying, I think, to anybody who's in cancer research these days, that we're in a major technological research revolution in biomedical research, where with single cell and spatial molecular technologies, we're now really able to capture for the first time, even in human patients, what's going on at a cellular to molecular scale. And then can we link that to therapeutic selection, with the idea being, can we design better therapies that are considering not only in the precision medicine paradigm of genetics and targeted agents of genetic drivers, but also in the immunotherapy context of combination therapeutics that are going to improve immune response and reduce immunosuppression. So along with these new technologies, if we want to get this understanding of the multi-scale system, of cellular and molecular landscapes, one of the things that I would really like to emphasize is that with the technologies, really part and parcel of them is also the invention of new computational methods. Because some of the fundamental cancer biology questions that we're trying to answer, for example, tumor evolution, what's the cellular architecture, what's the regulatory biology, these, yes, you can measure with the technology, but you need computational tools and novel methods in order to infer these, in many cases, from the high dimensional data and interpret it into something mechanistic. And, I'm grateful that there's, an emergence of many consortia now that are really starting to have this methodologic and software focus, as well as the biological focus, that really support all of our research moving forward. So, as Alice alluded to, I come to this background in thinking about these systems and tumor evolution and therapeutics, not in the context of a cancer biology background, but I come from a background in weather forecasting. And that really has informed how I think about computational biology, where one of the things that we can appreciate in weather forecasting is that it's the combination of our mechanistic understanding of the atmosphere with the emergence of high throughput data that really enables us to get to systems and understand, not only characterize where they are now, but can we predict where they are going to go in the future? And that's one of the things that I really would love, starting to think about as we try to understand cancer. Can we use the high throughput technologies to understand these systems? But then can we also try to go forward in time and start predicting the effects of different interceptions? So as we think about this, one of the things that we want to think about is what's the life cycle of the tumor, right? Because if we have this idea that if we can maybe get earlier, we're going to have better opportunities to treat patients while there's still a very active immune response. So we're thinking a lot in terms of pancreatic cancer and the early landscape of it. And one of the reasons this is such an interesting disease, in addition to it just being just a devastating disease when people are diagnosed, is that it's relatively mutationally silent. There's relatively few genetic drivers in this disease, and it has a very complex immunosuppressive microenvironment that evolves. And one of the things that's interesting is, in contrast to many solid tumors, a relatively small proportion of the sample is actually neoplastic epithelial cells. And you end up with this huge, dense composition of fibroblasts and immunosuppressive cells that protect the tumor as it's developing. So one of our goals as we think about this sort of cellular to molecular linkage of the data is, can we start to understand and tease apart the interactions between these molecular underpinnings and how they give rise to this immunosuppressive landscape? And then also from the therapeutic standpoint, does that give windows of opportunity where the patients might be more responsive to therapy and immunotherapy? So how do we get all of this, right, information when these cancers are, incredibly difficult to diagnose, right? So most of the samples that we get from patients, and most of the times patients present to clinic, is unfortunately at the time that they have late stage disease. But one of the things that we have in spatial data is that the spatial landscape of these tumors, even if you're at late stage disease with advanced cancer, you still have within the surgical specimen from the pancreas precancer and normal ducts within that same tumor. And if folks haven't seen this, there was an absolutely beautiful study that my colleague Laura Wood just put out in Nature that came out this week, like talking about the three-dimensional landscape and how you can see the evolution of these lesions by looking even in 3D in an advanced tumor. So in collaboration for my own group, in collaboration with Liz Jaffe's team, led by Wanjin Ho and Neha Zaidi, and a very talented graduate student, Melissa Lyman, we wanted to see if we could start to map the immune landscape of these tumors as they progress from normal to precancer, to see if we can start using spatial information as a surrogate for time in understanding the immunosuppressive landscape. So Juan led an imaging mass cytometry profiling of this cohort where we had several patients with matched, where we had normal chronic pancreatitis, Pannin lesions, and PDAC tumors all collected from the same individual. And what we observed, for example, here, looking at markers of T regulatory cells, is that these T regulatory cells increase across the spatial location as we get adjacent to these different lesions, suggesting that we can start to observe the transition in the immunosuppressive landscape as a function of space and use that as a surrogate for time. So this gives us a very powerful way to start looking at this data. The challenge with spatial proteomics technologies is that you have to kind of go in knowing what you're looking for, right? Because you come in and you have, you have to design it based on a marker panel. So this gave us a really great ability to look at what's happening in the T cell landscape, but not as much how is that interplaying with the transitions that we're observing in the epithelial cells that might give rise to the cancer. So in order to address this, we went to single-cell RNA-seq, which has the ability of giving us deeper resolution, and started to look at a reference atlas of pancreatic tumors to see if we can start mining the tumor and immune landscape at the advanced stages to see if we can relate that to the immunosuppressive responses that we see. So what's the first thing that you do when you get to a single-cell reference atlas? And this is work led by Ben Kinney-Koster and Jackie Zimmerman. The first thing you do is you get your single-cell data, you cluster it, and you identify cell types. And again, we observe that we see the malignant epithelial cells, dense CAFs now in this reference atlas. We see the lymphoid compartment and myeloid compartment coming out, so we can see that we're seeing some of the same populations. But of course, the challenge with clustering is that if this is all that we're trying to look at is these sort of cell types, arguably we have absolutely no advantage of doing this relative to a spatial proteomics panel where we went in looking for these types of cellular markers going in. So we really want to think about how can we go deeper in order to understand this landscape and the transitions that are occurring within the cells. So there's a great review by Wagner et al. And one of the things that they point out is that if we think about the power of single-cell transcriptomics, where we're measuring 20,000 genes in the human transcriptome across populations of cells, we can, yes, get out clustering information for cell types, like I was saying, but that this transcriptional information also leaves distinct signatures in the data that are associated with where the cells are, where they are phenotypically, where they are evolutionarily, where they are spatially, where they are in cell cycle. All of these things leave footprints on the transcriptional data. And our challenge is then, if we want to go beyond cell types to figure out what are the phenotypes of our data, is how do we infer from this data what these phenotypic programs are in an unsupervised way to learn from the data what the cellular processes are. So Genevieve Stein O'Brien had the realization that this type of transcriptional signature and trying to infer it maps perfectly to the mathematics of matrix factorization. So what do I mean by that? And I'm going to step away for a second, if you'll bear with me, from the world of pre-cancer and advanced cancer into this little cartoon example of 4 genes where I've shown their expression on the y-axis, and we can imagine the x-axis represents the value for each cell, and maybe the samples on the left are the precancers and the right are the advanced tumors. And what we can see by eye is that there's very clearly 2 patterns in the data, one of which that go up and down in the first group of precancers, and that's associated with genes 2, 3, and 4, and one of which going up and down in the second, and that's genes 1, 3, and 4. And so what we're able to do is then represent the data in terms of these patterns, which we hypothesize represent these phenotypes in our transcriptional signatures. And in contrast to clustering, where we aim to assign every gene or every cell to one group or another, this allows for genes and cells to be reused across multiple processes, reflecting the fact that, biology, for example, if you think about a cell cycle change, right, that might be shared across multiple phenotypes. So we've developed a particular approach for this that I won't spend too long on, called COGAPS, which is a Bayesian non-negative matrix factorization algorithm. And its primary advantage is that it's tailored to the sparsity of single cell data, which I'd be happy to get into later if people are interested in. But I'm going to focus a little bit more on the biology here. And one of the things that we wanted to see was, like I was saying, is how do we see these transitions in cell states between the normal epithelial cells and cancer cells in our single cell reference atlas? And what was in our cohort, if we looked at it, we had normal as well as tumor adjacent normal samples in our system. So we were hypothesizing that within our single cell data, some of these transitions might reflect the transitions that are associated with tumor progression, even though this is dissociated data where we don't necessarily have the pathology to validate that there's a precancer lesion in this. So we applied COGAPS to the epithelial cells in our reference atlas to see if we could start modeling these transitions and inferring the cellular phenotypes and molecular signatures that associated between them. And what we find is, if we look on the left in in the second pattern that was learned, it occurs within only the tumor samples. And that pattern is associated with changes in KRAS signaling and estrogen response, consistent with a proliferative program and growth factor receptor signaling that occurs in only the tumor cells. And this program seems to occur in a distinct set of cells relative to what we observe in another pattern, pattern 7, which contains both an allograft rejection, epithelial to mesenchymal transition, and inflammatory response, which interestingly has been associated with later stage pancreatic cancer. But in our system, we also observed the same type of a transition was occurring within the tumor adjacent normal cells, leading us to hypothesize that we might have some of these same programs in EMT transition occurring as a response to the microenvironment, even in normal cells during their transition. So we wanted to see, how do we actually map this and look at this, right? Is this actually something that's occurring in the microenvironment? Or is this because these tumor adjacent normal cells and these transition cells are actually pannins that we weren't able to uncover in the dissociated based single cell technology? So we were scratching our head a little bit and wanted to see, if we could map these on to pannins. The challenge with pannins, and this is no longer an issue now with technology, but when we started the study, it was, is that they're only diagnosed in FFPE. And the earliest spatial transcriptomics technologies were limited to OCT preserved samples, limiting our ability to profile these. So we reached out to 10X in a collaboration led by Luciani Kagahara and Laura Wood, and had the fortune to be beta testers for the early Visium FFPE technology in order to start mapping the transcriptional signatures across these stages. And Laura very generously went through the biobanks, and without us realizing what we asked her, collected a cohort in individuals with matched normal, low-grade, and high-grade pannins to enable us to evaluate these transcriptional changes in the pathway. So what do? We get this spatial transcriptomics data. You get the H&E, so you can find the different pannin regions. And then when you go to apply the same type of clustering that we would have applied for single-cell RNA-seq to start looking at cellular structures and cell subtypes, we observe in some cases, for example, the sample on the right, we do see clusters that are associated with distinct lesions. But far more often, what we see is that the transcriptional data and the clusters don't really match to any regions in the pathology. And this occurs because within the Visium's FFPE technology, you don't have true single cell resolution, right? So your spots are near single cell, but they combine multiple cells within them. And so then the trick becomes, how do you actually get the data and isolate it down if you don't have single cell resolution within the technology? So Ashley Keyman from Denny Wirtz's lab, now a PI in her own group, had developed a very elegant machine learning method called CODA that enables the use of H&E imaging in order to map, and then applies a machine learning classifier in order to map broad cell types from the imaging itself. And so what we realized was that we could apply the CODA approach to automatically annotate the different regions at a pixel level of what each cell was, and then overlay that onto the spatial transcriptomics data in order to get an estimate of the true cellular proportion in each region. And what that enables us to then do is then to purify our spots to those that are relatively pure, normal, low grade, or high grade, and we can start mapping transcriptional changes only across stages of the penin using this annotation from the imaging. So our goal, of course, is to go beyond looking at these single gene markers of the progression and look at this whole transcriptional signature that we observed that we're hypothesizing is related to the transition. And so one of the things that we had done previously was develop a machine learning method called transfer learning called ProjectR. And what that is if we imagine that these gene signatures that we've learned from our matrix factorization are the lenses through which we view a reference data set, for example, our single cell reference atlas of pancreatic tumors. If we have a new target data set, even from a different technology, in this case, our spatial transcriptomics data, what we can do is project these gene signatures that we've learned onto the reference data set. And instead of relying on mapping single genes to see where they occur, we can use whole programs and phenotypic programs and map where they occur on the tissue. So when we apply this transfer learning approach to our spatial transcriptomics data in the epithelial spots, we observe, consistent with our single cell data, a trade-off between this proliferative signaling and the inflammatory and EMT-like pattern in our data set. And now, because we have the pathology annotations at each spot for normal, low-grade, and high-grade lesions, what we can then do is also quantify how much are these changes occurring across stages of pancreatic carcinogenesis. And what we observe is that this proliferative signaling, we see increases across stages, whereas on the other hand, this EMT-like transition is, if anything, stable, arguably decreasing at later stage lesions, although with a relatively low p-value, leading us to hypothesize that what we're seeing in our data is that we are seeing proliferative signatures that are associated with the progression, but that this EMT and inflammatory signal is something that's probably more common due to the microenvironment of pancreatic cancer, and something that can occur across different disease stages and put a common selective pressure and force on the epithelial cells in the system. So one of the things that we also noticed when we looked at this is we wanted to then go and look at other cells in the microenvironment and see what was there. So with the Visium, and I apologize, the slide's a little bit blurry, we were able to map out different CAF signatures and observe the presence of CAFs. But unfortunately, we weren't able to look at really other cell types in the microenvironment due to the lack of single cell resolution of the technology. The immune cells and other cells were too big to look at. So we reached back out to 10x, and we were very lucky to, again, collaborate with them on the new Xenium for single cell imaging in order to do spatial transcriptomics of a true single cell resolution that enables us to get immune cell classification as well as the fibroblast classification. So we can then also get the epithelial cells and look at where the normal and pannins are. And what we can do is, because that it allows a set of custom probes, we were able to also design a custom panel to capture some of the proliferation signatures as well as some of the inflammatory signatures that we observed in our single cell reference atlas in order to map them. And what we observe when we look at a single cell resolution in the imaging data, at least in this one slide, and it holds across the broader cohort, is that we see, as we look at this region with the pannin, that the proliferation pattern appears to be more along the interior of the sample and sort of at the interior of the pannin stage, whereas the inflammatory signaling doesn't occur there, and if anything occurs further out in the edge. And again, we see the same mutual exclusivity. When we look deeper at the other cells in the microenvironment in the imaging, what we observe is that the fibroblasts are all around the edge of the tumor. And if we go back to our reference single cell data set, we observe that this inflammatory and EMT-like signature, interestingly, we only observe in one of the two cohorts of data that we analyzed, but it happens to be the one with more CAFs. And when we have a denser CAF composition, we're observing more correlation with this pattern. So taken together, this led us to hypothesize the possibility that CAFs are associated with EMT and inflammatory signaling and inducing that in epithelial cells. But of course, the computational biologist in me, when I see signal that only occurs in one of two data sets, I immediately get skeptical and wonder if it's a technical artifact. And so we really need some way to tease apart, is this just some weird computational finding that COGAPS is getting out of the data, or is there actually real biology here that's related to the type of signaling that we're seeing? So our colleagues, Jackie Zimmerman and Rick Burkhart, work led by Samantha Gwynn, a newly graduated graduate student, had developed a co-culture model of organoids and fibroblasts that we thought, could we start to see and use this culture model to evaluate whether the same types of changes that we're observing in our system are associated with fibroblasts in that system. So we have the human data set and patterns. Now, instead of applying the spatial transcriptomics data from human to human data, going from single cell to spatial molecular data, we can apply the same transfer learning approach from the human data set to transcriptional data from this organoid co-culture model to query in an isolated system, how much are we seeing these changes that are related to this? And of course, I wouldn't be giving this talk if it didn't work. So in the single cell data that we had from this co-culture model, we observed that this EMT and inflammatory signal is enriched in the single cell data. And if we look across a broader panel of organoid lines with bulk RNA-seq, we see that basically every single line is enriched for this EMT-like signal. And this is over and above this enrichment, I'm not showing it here, but is over and above the subtypes of whether they're classical or basal. And so we see this enrichment. So that we were very excited about this result. And so what do you do when you do a single cell reference atlas and a reference study? Is now at the end of every paper, right, you build this sort of cartoon network of how you think the system works, right? So we're now at a point where our hypothesis is that fibroblasts induce a mesenchymal transition in epithelial cells, whether they're tumor or normal. And as the tumor progresses, when the system is distant from fibroblasts, you see more of an epithelial-like tumor cell that's proliferating more, whereas the fibroblasts appear to be blocking cellular proliferation. So I wanted to go a little bit deeper, right, and see if we have these hypotheses, right, at the end of the day, we all build these network models of how we think biological systems work. How do we start going beyond building this network model to starting to test the implications of it. And, since I'm a computational biologist, focusing not on, biological mechanism in which pathways are there, but what are the implications of the cell-cell rules for cancer progression? So everything I've talked to up till now in the computational biology work that we focused on is really focused around data-driven inference, right? So we're thinking about, and I see so we're thinking about data-driven inference, where you have either, you're trying to figure out how the patterns are progressing. Where are they? What is their architecture, right? What's the regulatory networks between the cells? These are all things that we've been learning from the data. There's another form of computational modeling that I think is less talked about in the computational biology world, but more familiar to most mathematicians, such as myself, which is mechanistic models. So what are these? You can think of this if you go back to sort of your elementary, like physics class or high school physics. If, for example, you imagine that you are throwing a ball up in the air, right? We have the laws of gravity. You can write down a system of equations that describe the laws of gravity. And so if you know the velocity at which you're throwing a ball, you know exactly where it's going to fall forward in time. We can think about the same sort of thing in biology, right? If we know how cells are interacting, can we start to build out equations so that we can say, okay, fibroblast induces more EMT in an epithelial cell, no fibroblast, it's going to proliferate more, right? And we can start to think about those and how do we form equations. So, of course, that's easy for me to say. It's very hard to do in practice because for every single cell type and every single type of interaction you have, you would imagine that you have a different set of equations that you need to code up. So I approached my friend Paul Macklin about this, who's developed a beautiful math modeling software called PhysiCell. And I was like, Paul, how do we start to do this at scale where we can really integrate with genomics data? And what we have is that we have, in his software, what he has is that cells are each encoded. You get a series of virtual cells on your computer. They have a state and a process, and that encodes a set of equations. And those equations can either be based on what's going on within the cell, or what is the cell seeing in its microenvironment, or what's happening with neighboring cells. What Paul realized is that we could actually write out a grammar, or he could write out a grammar that would take in plain language words a series of templates. For example, cells proliferate, fibroblasts block proliferation, and use that and develop a grammar to parse those from plain language sentences into rules that could be encoded into default sets of equations. So this gave us the ability to start thinking, okay, if I have this little cartoon then of how cells are working, can I encode them into PhysiCell with these rules to model how cell-cell interactions are going to impact it? So Daniel Bergman led starting to think to abstract this. And what I'm going to show is we start out with epithelial normal cells that transition to tumor at some rate. And we have fibroblasts immediately surrounding them. Fibroblasts we're going to model as signaling to other epithelial cells through deposit of ECM components that then are recognized and signaling to the epithelial cells. So I'm going to play this movie forward in time. And what you're going to observe is initially the cells start to transition to be more mesenchymal-like, right? Mesenchymal tumor cells like we saw in our simulations. They're able to explore the space because they have greater motility. But then when we get regions that are more distant from fibroblasts, we start to observe formation of colonies that are more epithelial-like at the edge. So we wanted to then see not only, well, how does this work, but if this is the case, what are the implications of this in human tissue, right? Because it's very easy for me to draw these little cartoons of, oh, there's these few fibroblasts here, there's this mass of normal epithelial cells here, but we want to actually get at mapping what are the phenotypes that we observe in a real tumor. So Daniel went on to develop a beautiful piece of software that enables us to not only automatically take in the rules of the system that Paul has built, but also take in spatial transcriptomics and single cell data and automatically import that into the software so that you can then get from your single cell annotation, what are the list of cell types? You could then use that to define your rules and also get spatial coordinates of the tissue. So we applied that same fibroblast epithelial model to the spatial transcriptomics cohort that we had, now focusing on the regions of advanced PDAC. I see a question in the chat. Was it a reinforcement learning model? So it's not a reinforcement learning, it's an agent-based mathematical model. So what we now end up with is 2 different tissue samples. And now we can estimate, playing forward in time, what happens to these cells. So we observe during an initial period, the cells transition to be more mesenchymal-like, but at later stages, we start to see the development of these lesions that are more classical and more proliferative. And this is consistent with what's been observed in the literature, but not well explained, where the tumors, if you look at pancreatic tumors, the thought is that the mesenchymal phenotype is associated with metastasis. But then if you actually look at the metastatic lesions, they become more classical and epithelial-like. And what this observation in these simulations would suggest is that the mesenchymal state is a transitory state induced by fibroblasts, but then once the system's able to go free, then the cells start to proliferate, they transition during the invasive state because they're no longer seeing fibroblasts to being more proliferative and more epithelial-like, exactly like's been observed in genomic studies. So this gives us a really nice way to start thinking about modeling cancer progression and carcinogenesis, right? How do these tumors grow? How do they start to evolve over time? The other thing that we can start to think about now that we have this is computationally, we can perturb different parts of the cellular environment and start thinking about, if we perturb the cellular environment, then how do we start to get at therapeutics, right? So we could imagine knocking out a cell type or knocking out a gene and developing a virtual system to start testing the implications of clinical trials and therapeutics. So how are we going to start mapping the rules of clinical trials and immunotherapeutics? So my collaborators, Liz Jaffe and Lei Zhang, have a neoadjuvant clinical trial platform that they've developed to elucidate therapeutic response and resistance mechanisms in pancreatic cancer, where what they're doing in this trial is adding different combinations of immune checkpoint inhibitors, immunotherapies, starting with the vaccine strategy alone, and then the introduction of the vaccine and nivolumab for an immunotherapy, to start looking at what are the implications of therapy and start treating patients. And because these are done on a neoadjuvant stage, you then get out surgical biospecimens that you can start looking at to look at immunotherapy response, that you can start looking at to look at how, sorry, forgive my language, how the immune cells change in response to the different therapeutic groups. So this was one of the things that I think we don't appreciate is how long these trials take to get up and running. And so when Lei and Liz started this platform trial to start looking at this, it predated single cell technologies, but we were fortunate that they were able to do a flow sort of different immune cell populations and do bulk RNA-seq in order to start looking at those in an analysis led by Joe Tandarella to start looking at the gene expression changes as well as TCR clonality and BCR clonality as it relates to the different combination therapeutics. So what we observe when we look at this data is that when we look at vaccine relative to immune checkpoint inhibitor, the dominant signal that we see is that vaccine alone has a large percent of naive CD4 T cells that become less naive with the combination of checkpoint. When we extract TCR repertoire data from these same samples from the T cells and look at survival, unfortunately, we observe that CD4 response has nothing to do with response, but CD8 T cell clonal expansion is the dominant signature due to response. So that gives us a sense that we're starting to see that the checkpoint inhibitor in pancreatic is activating the CD4 T cells, although we would like to see more of a CD8 response. We then went on to do differential expression, just a relatively simple differential expression and pathway analysis, comparing what happened in the trial arms. Consistent with the observation that we had less naive CD4 T cells, we see more increased chemokine receptor binding. And interestingly, the other pathways we had to scratch our head around a little bit more, where we saw lymphocyte chemotaxis signaling in the CD4 T cells, extracellular matrix in the CD8 T cells, and neutrophil degranulation observed in the monocytic populations with immunotherapy response. So again, we were scratching our heads a little bit about the relationship between all these pathways and started to form the hypothesis, well, what if what's happening is that when the neutrophils are degranulating, they're changing the structure of the extracellular matrix, which then could have a hindering effect or an effect on CD8 T cell motility that's counteracted by CD4 T cells releasing chemokine to attract CD8 T cells. So what are the implications of that? So we simplified out this model, just imagining we have ECM components and leaving out the neutrophils for now, and built a PhysiCell agent-based model of this system around this in order to start looking at the implications of this signaling. So what we have is a mass of tumors that have a ton of macrophages in them, consistent with the immunosuppressive environment of pancreatic cancer. And we imagine around the edges, as we're introducing vaccine, that the lymphocytes are starting to traffic in. And what we observe when we run this model is that the lymphocytes immediately form aggregates. So Jeanette didn't show me this data for the longest time because she was worried about the model and not getting any response, until I pointed out to her that this is pancreatic cancer and we're not getting large patient responses or tumor regressions. But what has been observed in the immunotherapy is the formation of lymphoid aggregates. So this gives us great hope that we can sort of start to model this type of system. We then wanted to see, could we go deeper and not only model where we have therapeutics, but how can we start to model the effects of new therapeutics, right? Can we start to predict what new therapeutics that haven't been seen in the system are going to do? So one of the next therapies that Liz and Leigh were looking at in their clinical trial is the addition of a urelumab as a CD137 agonist. And we wanted to see within the tumor microenvironment of untreated tumors, can we start to tease apart the signaling changes that we could hypothesize would occur by looking at immune cells based on their CD137 expression? So we went back to our reference single-cell RNA-seq atlas, pulled out the immune cells now, and now we're able to classify our effector CD8 T cells based on the expression of TNFR-SF9, which encodes for CD137. And we wanted to see, what are the implications of these cells? What are they doing in the microenvironment, if we have these cells that are high for CD137? So one of the things our colleagues Jennifer Alicieff and Chris Cherry developed is a ligand receptor network inference tool that enables then the modeling of how different cells communicate by looking at correlation between ligand and receptor between cell types. And what Jacob observed was that when he looked at the cells that were expressing CD137, they appeared to signal to neutrophils through increased interferon gamma expression, which Jeanette hypothesized in our model would occur by CD137 high CD8 T cells having an independent chemokine release from the CD8 cells, or from the CD4 cells. So what are the implications of this on the structure that we see in the trial? Again, if we have vaccine alone, we see the formation of aggregates. If we add in immune checkpoint inhibitor, what we can now observe is we still get the same aggregates, but we get some tumor cell killing around the edges of the tumor. If we look at urelumab alone in our simulations, these aggregates now infiltrate inside of the tumor, and the triple combination enables the infiltration and killing more within the depth of the tumor. So our next phase, as we're doing this right now, this is all an amazing theoretical framework. Our next question is going to become, how do we know if any of this is real and related to patient data? So we're fortunate that Liz and Lei have follow-up single-cell RNA-seq on this triple combination, as well as the original vaccine and checkpoint alone. There we do see, sort of consistent with this, enhanced CD8 TCR clonal expansion from RMC, which would be consistent with the type of killing that we observe in our simulations. But it'll, I think, be sort of the next bit of the lab of how do we think about really starting to merge these and get the robust validation of the results. And so with that, I'd like to make a call for us as a community to stop thinking about precision medicine and start thinking about predictive medicine, where we're really starting to understand not only what cell types are there, but what are the implications of these cell types in terms of cell-cell interactions and how the tumor is going to change over time as it responds to therapy due to these more complex interactions. And for this, we're really going to need to integrate all levels of patient data with mechanistic mathematical models in order to start getting at these mechanistic predictive interpretation. I think the field's also going to have to see a little bit how much mechanism is going to play out relative to artificial intelligence methods for prediction. I think that's going to be another whole area of research as well to sort of cross-compare them. And with that, I'd also like to make a call not only for this type of work, in terms of the modeling, but also thinking about this in terms of really deep team science and convergence research between investigators from different disciplines to start to fully explore this system. And with that, I'd like to, of course, thank all the members of the lab who contributed to this work, as well as collaborators and funders, and I'm happy to take questions.
Thank you. Thank you so much, Elana. This was absolutely fantastic. So as we said earlier, please, if you have any question, do put it in the Q&A so we can see them. That would be the easiest for us. But maybe in the meantime, let me start to asking you something about the pancreatic cancer work. So there is, I guess, maybe debatable, but fairly accepted knowledge that there are different types of fibroblasts, right? Are you looking at that? Are you able to separate them and tease that apart? Is that too high?
Yeah, that's very much where we're headed next with some of the modeling. So we can tease that apart in the spatial transcriptomics. And we definitely saw the, AP-CAFs present there, as well as the different MyCAFs and ICAFs. We haven't seen as big signaling changes as I would have expected from the ligand receptor network inference between the CAF subtypes. As I don't know if we're missing something in the signal or what's going on there, but the changes haven't been as pronounced as I would have expected in the signaling mechanisms. But that is very much where we're thinking is like, what are the implications of those on the microenvironment and the types of phenotypes that we see?
And are you also looking at other cancer types? Because there is quite a few that have similar progression. patterns?
Yeah. So we've done this also in liver cancer. We've built sort of parallel models in liver. And, you know, again, I think anything that has this mechanism, right, it could work on. So the software is generic. Most of our focus has been pancreatic, but, you know, it could be applied across cancer subtypes. And we've looked a bit in breast as well.
Everyone else is shy. So I'll ask you just one more question. So I find the neutrophil, the granulation, you know, very interesting, right? So the signaling and the changes in or how they may be recruited there, etc. That makes sense. But the granulating, why are they the granulating?
I don't think we know, right? Like, yeah, I think that that's going to be a whole other thing of really looking into what's going on in the neutrophils in the population. I'm hoping that that's where some of the spatial technologies are really going to help us, because we don't have the dissociative based techniques. The neutrophils tend to explode.
Okay.
So you don't end up able to look at them. So I'm hoping we can get at more of the signatures that are there. I've heard conflicting information from my clinical colleagues as to what the mechanism is, and like, is it changing the ECM and having something to do with that with the neutrophils nets? Or is it that you see more neutrophils in patients that have worse outcomes because the neutrophils just form at later stages when the patients are doing really badly? And so is it just related to something, you know, is it correlative or is it causative? I don't know if we know that yet.
And is there a reason why you don't model those within your, or you don't include these within your models?
I think that'll be a next stage. I think it's because we don't have data on them right now. Like we don't have the single cell data to validate. And it was a level of complexity that we didn't have yet. So we're aiming to go deeper into the ECM because it's easier for us to generate data of ECM alignment and things like that first. So that's where we're heading in the lab right now. But I think understanding the neutrophils and how to model them is definitely of great interest.
Yeah, and how that correlates to, I mean, it's such an innate, you know, immunity type response of them. Doing that. I'm sorry, there was something in the chat. Yeah. Okay, no, that's a separate thing. All right. Well, if we don't have any
That's
other
a quiet bunch.
I know it's a quiet bunch today. But if we don't have any other question, then please join me in thanking Dr. Fertig again for this fantastic seminar. We appreciate, you know, having you, albeit virtually, and hopefully we can get you on campus soon enough to visit us again.
Thank you so much for having me. It's my pleasure.
Bye. Bye, everybody. Thank you so much. Bye-bye.